I'm a sweet potato pathologist, and I'm in the lab that actually delivers the clean and meristem tested, virus tested plants to this lab. That's a huge part of what we do in the lab, making sure we have cleaned up the breeding lines that we get from Dr. Labonte, and we remove the viruses and test to see if we actually remove the viruses and then keep them in tissue culture and deliver them here. The update I want to give today is about what we actually do beyond the lab, right? Because it's nice and fun when we're in the lab and we do all those molecular tests. It takes about a year to clean up and test everything. But that would not be sufficient enough for a grower, a seed grower in the field that has acres and acres of land, of plants growing, and wants to know if they are able to keep their plants clean as well, right? So we know, yes, reduce reinfection, but what about making sure that you don't see any infected plants? Remove the inoculum, right? So the potaviruses have beautiful symptoms when they infect sweet potato, and you can actually see it. We know them, this right combination of purple ring spots that they form, but that is what we can see. For a large part of the time, in the beginning when we planted these sweet potato plants can be infected, but not yet symptomatic. So there are no symptoms yet, but they are infected. So how do we go about that? How do we take those in the field where you have acreage and acreages grown? That's part of Clean Seed project that we're working on. It's a graduate student in the lab. I encourage you as well to read the newsletter, because Clayton Blake is in there. And we use this spectral imaging camera to see if we can actually detect viruses that are in the plant, but no symptoms are developed yet, right? So this camera is a nice and expensive little camera.
It's $45,000. I don't have to drop it. Is it a paper spectrum? It is.
Right. And so we used a greenhouse study, controlled study, where we infected plants. We had healthy and infected plants with just one virus, and we imaged them at specific times before the symptoms were actually visual, but we knew that they were infected. We tested that before and after we took images. And we took images of both leaves and whole plants of 2 cultivars, Beauregard and Orleans, as well as the indicator plant, Ipomea setosa, to see if there is a way to detect it beyond human vision. A lot of AI, machine learning. Clayton is great at it, all those the coding that he needed to do in Python and all. And he was able to distinguish infected from healthy plants, not yet visible, with one of the machine learning methods, 85% of the time, 85% accuracy. That's actually really great. Just imagine if you would scout your plot, it's not visual yet, that would be 0% accuracy that you would be able to get it out. And now he got it up to 85% accuracy. So that's nice. But that's a controlled study. We have a standardized height that we have the camera in. We have the lights that are beautiful. We have just one virus in there. In the field, we have mixed infections, different types of infections of those 4 potaviruses that we're focusing on. And light settings are different. And it's an acreage. It takes, I think Clayton told me it takes about 10 minutes, a minute to image once it's all set up. That's a plant, imaging a plant and then the leaves. So to figure out if we can upscale it, we want to see, can we use either a robot that goes in the field, or can we use a drone to help the growers, the seed growers, to better detect this? So that is the next step, of course. That's on virus, making sure if we can optimize our virus testing. So 2 more updates. As I said, plant pathology, sweet potato pathology. So we also work on alternatives to Botran, dichloran. This is a fungicide that's been used for over 40 years in the sweet potato production, since the 70s, in the seed beds as well as in the packing houses. So we started with it, and Goan Industry is not producing Botran anymore since 2023. So whatever is out there is what can be used. So that means we really need to have answers immediately. What can be used instead of?
So what diseases was Botran targeting, just for the sake of information?
Sclerotial blight.
And you're talking about seed beds here.
Right, in seed beds, it's sclerotial blight and scurf. So Agrotilia, that's the official name, Agrotilia ralphsii, Sclerotium ralphsii, and Moniliocydis infuscans. Those are the two that Botran is active to, it's registered for in seed beds. In packing houses, it's different. The update today is really about the seed bed study we did this summer in great collaboration with Black Gold here and at St. Joe's and in Baton Rouge, where we had Botran, Murtec, all fungicides that are used in sweet potato that we tested for their efficacy in seed beds as a alternative for Botran, whether or not it has similar effects or better effects or not, right? We used 4 cultivars over those different locations, and what we're doing now, and we took soil samples, we used the same method of application that Black Gold uses. Botran, before you put the seed potato in, Botran and Murtec, I should say, you put the seed and then you spray again, you cover it up, and you cover it up with soil and plastic. We did that. We did those 2 chemicals separately, and we used 2 additional fungicides, Scholar and Satori, thiabenazole fludeoxonil. So to make sure we can have good information, we're now testing if we can detect and at what level we can detect those pathogens that cause these diseases in the soil. And in December, we will be able to give you an update on the efficacy. But beyond that, because if we're going to use additional fungicides, we also want to see if they have an effect on other soil pathogens that are problematic in sweet potato. We don't have here in Louisiana black rot. But what if, right? Because some in North Carolina, in other states, we know that they do have black rot in the seed beds. So will these fungicides that we're testing have an effect on those as well? And then at some point, we want to see if there are other fungicides out there that have not been tested on sweet potato. That's a later stage, and see what those effects would be. The final part is a disease, or it's well, I shouldn't say disease. It's stress, but it's abiotic, right? So we heard Dr. Philorden talk about drought stress, and we're working on flooding as well. And we want to have an idea of what can be done or what a flood actually does to the plant, because growers want to know, producers want to know, once they plant and there's a late season flood, how does it affect the sweet potato? Are varieties differently responding to it? We see with NPK, with all the drought information, that there is a varietal effect. But what is it in relation to flooding? Apart from that, can we use something on top of the sweet potato to see if we can reduce, once the plants are flooded, if we can reduce some soft rot? Because that is what happens, right? If these plants are flooded, depends on the duration, you can have additional, other than the post-harvest diseases that we are familiar with, you can now have different types of soft rot developing. That's a lot of work that Dr. Clark and Dr. Labonte did before I was in Baton Rouge. One of their studies was to actually inundate roots in mason jars and just see the effect, right? What happens, what pathogens do we have? So we're going to expand on that. And so we're going to grow 50 days, 60-day-old plants at some point, and then flood them in these pots. Okay. This is all being done here. Thank you, Dr. Gregory.
And they'll spend 5 days completely saturated.
Inundated. to have an idea, and then we compare them, same cultivars, and compare the duration, and remove them, also check which ones of these cultivars recover or not. You know, if you have a flood for a specific duration and after that it dries out, what happens? So we're looking at that, and we have another study in Baton Rouge that's going to be almost similar, but that will be a later season flood, so 100 or 120 day plants, and then test post-harvest effect. You know, so those post-harvest pathogens that we know, how are they differently affecting sweet potato after a late season flood? And so this is where you integrate information, right? Because now that Clayton has developed this whole machine learning imaging using hyperspectral imaging, what if we can use the same method that he has developed and see if there is an above ground stress that we can detect using hyperspectral imaging that can also tell us something about the stress, the abiotic stress that these sweet potato plants are undergoing. So that's it's just an update on the work we're doing. I mean, I work with, I think, everybody in the sweet potato pathology team here at LSU and above, and we get funded by the Sweet Potato Commission, as well as my department, the Plant Pathology and Crop Physiology department, and we get in-kind, collaboration with Black Gold, so that's all really important to mention. Thank you all.